right? Well, I like that. No new lab work tomorrow. Two things tomorrow is you'll be getting back your lab exams, and we'll go over the lab exams. The other thing that you can do tomorrow, if you haven't already done it, is remember that the answers to those, I think, 4 questions relating to the skin, nose, and throat are due by end of day tomorrow. And so it'll only probably take about half the lab period if they have to go through the lab exam. So then you have the remainder of the period to work on that if you want to. We're going to really shift gears next week. We're going to start working on metabolic tests for your unknown, and this is where you're going to start really piling up information that's going to allow you to identify your unknown. Those of you that have been complaining that your unknown is really boring, it's a little gram-negative rod that doesn't seem to have any kind of discernible structures or anything that are interesting, you'll get a lot of interesting information about the organism as a result of analyzing its metabolism. So fermentation tests, all kinds of information is going to be accumulated. So in the next couple of weeks, you're really going to get the stuff you'll need to do to complete the identification of your unknown. So make sure for next week that you have a good viable culture. I would probably, maybe tomorrow, that would be a good time to subculture your unknown if you haven't done it for a while, and go ahead and make sure you've got a good slide of that. Okay? Okay, we've spent some time talking about 3 or 4 major classes of molecules that can be used in therapy of individuals suffering from infectious disease. The antibiotics that affect cell wall synthesis, and the antibiotics that affect protein synthesis. We also made some brief reference to some drugs that can interfere with either DNA replication or transcription of DNA into RNA. And then last time we talked about a few of the antiviral drugs, concentrating on HIV as an example, and looking at the HIV life cycle and seeing there are a number of places where vulnerabilities can be exploited that are unique to the virus replication cycle, but don't have counterparts in the host cell's replication cycle. So there are some interesting and effective antiviral drugs. The problem with that is that those drugs have got to be prescribed very shortly after symptoms are first noticed. Otherwise, if you wait too long, the infection has just become too widespread, and those drugs just aren't going to be effective. If you reach that particular point, then you've got really one alternative, and that is hope that your immune system kicks in and eliminates the threat. And that's where rest and hydration and things like that come into play, because there are things that you can do that will compromise your immune system. And one of the things that compromises your immune system is lack of sleep. It's really not much of a surprise that many of you will get sick during finals week, because your immune system will take a crash because you're not sleeping. And also, we'll see later on that stress can have a real important role in your immune system. Stress hormones actually inhibit various key immune processes. So many times you struggle through finals week, and then as soon as finals is over, you get sick. It's not surprising. Antibiotics have been a wonder drug. I think I mentioned that in the pre-antibiotic era, about 1/3 of people, of children, died as a result of bacterial infections. That's one of the reasons why families tended to be larger in previous generations. Antibiotics, however, are losing their effectiveness because organisms are developing resistance to antibiotics. And that's one of the things we'll spend some time talking about. Some public health people firmly believe that really this is the next really global medical crisis, is the development of widespread, multi-drug, antibiotic-resistant strains of bacteria. And if we don't keep up with the production of new antibiotics and find new strategies, then we're going to lose that war. And if we go back to the pre-antibiotic era, it's going to be pretty nasty. Where's a place where you are probably more likely than other places to acquire an infection? The hospital. Okay. Have you ever heard of this term, nosocomial infection? It's also called an HAI, hospital-acquired infection. Statistics indicate that you've got about a 5 to 10% chance, if you're hospitalized, of developing a hospital-acquired infection. And unfortunately, many of those, about 60% of those infections, which number about 2 million per year, about 60% of those are drug-resistant forms of bacteria. So MRSA, for example. This is thought to contribute to greater than 80,000 deaths per year. That's significant. Okay. We freak out if we have an airline accident, for example, and one jumbo jetliner carrying maybe 600 people goes down and crashes. But this idea of 80,000 people dying per year from hospital-acquired infections really doesn't get much publicity. It should. It should be a major public health initiative to try to prevent and decrease this particular number significantly. And it's hard. There's also costs associated with it. Even if you don't die, if you get a hospital-acquired infection, your length of stay in the hospital is going to be extended fairly significantly. Because many times you'll have to be on intravenous antibiotics, for example. And that adds to hospital costs, that adds to healthcare costs. Conservative estimates indicate hospital-acquired infections add about $10 billion a year to healthcare costs. It's not insignificant. So one of the things we need to look at a little bit, particularly in the context of these resistant pathogens, is how did we get there? Okay? What's contributed to the development of these resistant forms of pathogens that no longer respond to the wonder drugs that we thought at one time were going to eradicate disease? Well, one of the big things that contributes to this is overuse of antibiotics. Remember that antibiotics were discovered in the 1928, but really didn't gain much of a foothold until World War II. They really didn't start being sold to the general public until following World War II. So I grew up, born in '50, in kind of the dawn of the antibiotic era. Antibiotics were so pervasive at that time that they were put in all kinds of things with the idea that, well, let's expose people to these antibiotics, and then if they happen to get, exposed to some potentially infectious agent, that it's going to protect them from it. Okay? So, prophylactic use, preventative use, was really kind of a rule of thumb. And antibiotics used to be put in all kinds of weird things. There were antibiotics in chewing gum. Okay? There were antibiotics in toothpaste and mouthwash. And the idea is that if a little bit of these drugs are good, a lot of them are probably better. What is the first initiating event in the development of resistance of a microorganism to an antibiotic? What really has to happen first? There has to be a mutation. Okay? And we'll talk about what the nature of some of these mutations are. and how they might ultimately result in resistance. But a random mutation, okay? What do mutations usually do to an organism's fitness? A random mutation. There's usually some kind of cost associated with mutation. And oftentimes the mutant forms are not as fit as the wild type forms, unless what's present. So mutation that results in a gene that codes for a different protein that confers antibiotic resistance. In the absence of the antibiotic, you might be less fit, and that mutation might be a disadvantage. You're not as competitive with your other members of the population if you're the mutant, unless the antibiotic is present. In the presence of the antibiotic now, that mutation becomes an advantage. And individuals that do not have that mutation all of a sudden become at a disadvantage. So what do antibiotics do? They change the rules of the game. They change the survival rules. The antibiotic is an agent of selection. It's a natural selection agent. So in the presence of antibiotics over a relatively short period of time, we can go from a population that has a very low frequency of a mutation in a particular gene that confers resistance. Why is the frequency low? Because that particular mutation decreases the fitness of the microorganisms. But if you start exposing that population to antibiotics, the rules of selection change now. And even though that mutation gives you less fitness in some aspect, it confers a tremendous advantage in the presence of the antibiotic. So the antibiotic tends to select against susceptible forms and select for resistant forms. So really, the development of antibiotic resistance truly is evolution. How do we define evolution? That's in comments coming up sometime, don't you guys? What is the definition of evolution? Do individuals evolve? We talk about evolution, what kind of group are we talking about? Not the individual, but a population, right? And something's changing within that population. What's changing? Alleles, the frequency of genes. Okay, so a very simple definition of evolution is a change in gene frequency in a population over time. That's what's been happening in the period of time since the 1950s, and actually a little bit before that. The frequency of alleles that confer antibiotic resistance is increasing within bacterial populations. The bacterial populations are evolving. In response to what? In response to the selective agents, in this case the antibiotics. One of the common misconceptions about resistance is that the antibiotics cause the resistance. No, mutations cause the resistance. Antibiotics select for resistant forms. You may know what the definition of natural selection is? A concise definition? One way you can define natural selection is by saying it is differential reproduction. Some organisms survive better than others. We sometimes refer to it as survival of the fittest. Okay? So when someone tells you that there is no scientific, observable information that allows us to say that evolution occurs, it's wrong. Okay? The evolution of bacterial resistance is a clear example of evolution in action. And why does that evolution happen so quickly in bacteria compared to other organisms like us? They reproduce faster. Okay, their generation time is very short, and in a relatively short period of time, we can select generation after generation for the resistant forms. It's kind of scary. This one's a little bit hard to read, so I will refer to it, just have you look at it in your PowerPoint notes. But this is the percentage of Streptococcus pneumoniae strains showing penicillin resistance. They're saying here that the introduction of penicillin was in 1940. Actually, now they have actually gone back to isolates that they had pre-1940, and there actually were resistant strains of Streptococcus pneumonia to penicillin even before penicillin was really put out in the market. Why? Because some strains developed this random mutation. But what did the introduction of penicillin do? Selected for those particular resistant strains. So we didn't see the real first bump in resistance until 1979 in Spain. And we had a relatively small amount of resistance in the population. But over a period of time, in the 1990s, the 2000s, those bars indicate greater percentage of resistance. So if you look at Korea right now, between 80 and 90% of the strains are resistant to penicillin. It's scary how fast this resistance can develop and spread over a relatively short period of time. And it gives us pause to think about that, because even once we recognize the pattern of resistance developing, drug development, if that's how you're going to tackle resistance by developing new drugs, takes so long that it's really hard to keep track and keep up with the rate of resistance and keep ahead of it by developing new drugs. Now, there are also other ways that you can try to get a hold on this problem of antibiotic resistant strains. And one thing is develop new drugs. One of the things that the introduction of antibiotics did was it really discouraged preventative methods. Okay? So when there was no antibiotic like tetracycline to treat syphilis, then public health education about avoidance of getting that particular disease was quite important. But it was a hassle. Okay? Condoms, for example. Develop a drug. You don't have to worry about that particular avoidance now. You just take a pill if you get diagnosed and you're good to go. So there's going to have to be some kind of retrenchment, if you like, and going back to trying to figure out how we can prevent these diseases from occurring as opposed to just treating them after the fact. That's going to have to be part of our strategy if we're going to keep ahead of this particular game. Okay, so there has to be a mutation that occurs in order for resistance to develop. Once that mutation has occurred, the scary thing about antibiotic resistance is that genes can be transferred from one organism to another, and an organism can acquire resistance from another bacteria. That's particularly true if the resistance genes are on plasmids. Remember that bacterial cells can interchange genetic information. So if these are two bacterial cells, and this is the bacterial chromosome, and this is a plasmid, if this cell copies its plasmid, that copy can go through this pilus or conjugation tube, and a donor or a recipient cell can acquire that particular plasmid, and whatever genes are on that plasmid potentially then can be expressed. And if there are genes for antibiotic resistance, those are oftentimes called R plasmids for resistance plasmids. This particular resistance can spread very quickly and very effectively. So conjugation is one way that can happen. Another way that resistance can be spread is through the process of transduction. Does anybody remember what transduction is? It's not direct gene-to-gene exchange from bacteria to bacteria. There's a vector involved. It transfers genes from one bacteria to another, but they're not in direct physical contact. The cells are. What's the vector? Virus. Okay. What's really quite interesting is we keep trying to superimpose on bacteria, for probably good reasons, this idea of the concept of species. Okay? And I'm going to ask you to identify your unknown to genus and species. But again, remember, the concept of species really breaks down in the microbial world compared to the eukaryotic world. What is the criteria for species in plants, animals, other eukaryotic organisms? What do you have to be able to do if you're considered members of the same species? Breed, reproduce, and produce viable offspring. Okay? Well, bacteria can be very promiscuous about transferring genes, especially via conjugation, from one species to another. So E. coli, for example, can both transmit and receive genes from a number of different bacterial species. E. coli, do you remember gram-positive or gram-negative? E. coli is a short gram-negative rod. It's one of those enteric bacteria. Okay? Well, Klebsiella is a different species. E. coli and Klebsiella can exchange genes. It's also a gram-negative. Serratia is a gram-negative. Neisseria is a gram-negative cocci. Another gram-negative. Shigella, another gram-negative. Another gram-negative. Staph and Bacillus, are they gram-negative? No, they're gram-positive. Okay? Bacteroides is another gram-positive. So the idea that what we think is kind of fundamentally different groups, gram-negative and gram-positives, can exchange genes, that's pretty amazing. Okay? And then E. coli can exchange genes with Pseudomonas, and then Pseudomonas has got all of these opportunities to exchange genes. What's really scary about this is that many of these organisms, like E. coli, are gut bacteria. And so if you've got these things living in your gut, and they can exchange genes with other resistant forms, you can run into some real problems. Most strains of E. coli are non-pathogenic. But about a decade ago, a very pathogenic form of E. coli, called hemorrhagic E. coli, popped up. And if that became part of your normal gut inhabitant, like exchanging genes with resistant forms, that was a problem because it was lethal in many cases. So, what are some ways in which mutations can result in the development of resistance? Well, probably one of the best known mechanisms are drug inactivation mechanisms. We've used penicillin as an example of an effective antibiotic inhibiting cell wall synthesis. But mutations resulted in enzymes being produced called penicillinases that could actually degrade the penicillin molecule. Penicillinases are part of a larger class of molecules called beta-lactamases. Remember the penicillins and cephalosporins both have that beta-lactam ring in their beta-lactam nucleus. And what these particular enzymes do is they hydrolyze that beta-lactam ring. So up here we have penicillin, and here's the beta-lactam ring. Under the influence of the enzyme penicillinase, that beta-lactam ring is inactivated. Okay. And you remember what the penicillin is doing? Why does it normally? Because it binds to what we call penicillin binding proteins, enzymes like transglycosylase and transferase that are normally required to build new peptidoglycan. So if this ring is hydrolyzed, this molecule will no longer bind to those penicillin binding proteins. Well, this particular enzyme, beta-lactamase, could have resulted from just a random mutation. But do you think organisms that had this particular mutation would be very fit? Maybe, maybe not. Mutation oftentimes has its costs, has its consequences. Why would this particular drug have been selected for, potentially? There's a genus of bacteria called Streptomyces, the gram-positive soil bacterium, that produces many antibiotics, including some forms of penicillin. What's the risk of being an organism that produces penicillin? Killing yourself, right? Okay, you could inhibit your own cell wall synthesis. So might the production of a beta-lactamase that kind of destroys internal penicillin be an advantage? Okay. Well, if an organism like Streptomyces develops this particular mutation, then they could potentially transfer that gene to other organisms and spread that particular resistance. There's another way you can inactivate drugs besides degrading them and breaking them down. Bacteria have developed enzymes that can chemically modify drugs like antibiotics. How do they do that? They can actually add functional groups to them. Phosphorylate an antibiotic, stick a phosphate on it. Hydroxylate it, stick a hydroxyl on it. Methylate it, stick a methyl group on it. Could that potentially have similar consequences to breaking this bond? Yeah. Okay. If you change the structure of the antibiotic, then it might not be effective. How might organisms respond to that? Or how might we respond to that as drug manufacturers? Chemically modify the drugs so they no longer will be recognized by this enzyme. That's what we did when we developed methicillin. We tweaked the structure of penicillin so it no longer was recognized by this penicillinase. Tweak the structure of drugs so they're no longer recognized by these chemically modifying bacterial drugs. You see how it's kind of a back and forth, almost war? Bacteria develop a strategy, and then drug manufacturers have to try to respond to that strategy by somehow changing the molecule and making it not respond to that particular resistance strategy. It's pretty complicated. Another public health scare that is kind of concerning is there is a new beta-lactamase that has been recently identified called NDM1. This was first discovered in New Delhi. And this is a beta-lactamase variant that bacteria are now producing. And formerly resistant forms of penicillin, for example, no longer are resistant to this new beta-lactamase. Okay, so normal forms of beta-lactamase hydrolyze the ring. So what do? We modify the antibiotic so that the beta-lactamase can't recognize it. Bacteria responded by developing, in this case, NDM1, and that new lactamase can now recognize a modified drug like penicillin. This is one they're keeping a really close eye on, because this really could cause a whole shift in the strategy of drug companies having to develop new drugs to respond to this particular threat. So that's one strategy, is drug inactivation. There are other strategies as well that can result in the development of resistance. Another one is things that decrease the permeability of the drug to the cell that's being treated. Okay? Drugs have got to get into cells to be effective. So those bacteria can develop mutations, for example, that results in less chance of the drug getting into the bacterium. They will be more resistant to the effects of that particular drug. There typically has to be some kind of a binding of the drug to the bacterial cell in order for the drug to get into the cell. And that binding is usually facilitated by the drug binding to a particular receptor. So what happens if the bacteria has a mutation that changes its receptors a little bit? Maybe there's a particular carrier in the bacterial membrane, or maybe in the LPS layer, one of those porins, that the drug binds to get delivered inside the bacterial cell. What if there's a mutation in the gene that codes for those porins? Now, if the drug can't get inside, the bacterium will become more resistant. I mentioned VRE previously. Stands for vancomycin-resistant enterococcus. This is a resistant form of pathogen, enterococcus, or bacteria that inhabit the gut. And they have developed a resistance to vancomycin. One of the first steps in treating bacterial cells and killing them with vancomycin is the vancomycin has to get inside the cell to be effective. Mutations have resulted in decreased permeability of those cells to that particular drug. And that has resulted in this VRE strain now. Well, let's say that a drug does get inside of the cell. Does that mean that it automatically is going to have action and kill the cell? No, not if the cell pumps it back out again. So there are a number of very sophisticated drug pumps that work in the cell membranes of bacterial cells that just pump the stuff back out once it enters. So it doesn't matter if the bacteria is permeable to the drug or not. It's kind of like a leaky boat. Water leaks in, you bail it back out again. Now, probably what's happened is that existing metabolic pumps in the membrane that have other functions have mutated so that they can also pump drugs as well. Some of those drug pumps will pump out multiple different types of drugs. So there's not necessarily just one pump that works on one drug. When we talked about biofilms at the beginning of the semester, I said that a significant number, I think I threw out the word like 2/3 or something, of human infections are the result of bacteria that are part of biofilms. In biofilms, the bacteria start communicating with one another through cell signaling, and genes that have been inactive when the microorganism isn't part of the biofilm oftentimes get activated once they're part of this more complex community. This is one of the things that typically happens in biofilms, is that these drug pumps become activated and become much more effective at pumping out drugs. Now, number one, if you're part of a biofilm, it's harder for the drug to penetrate the biofilm to begin with. But even if it does, and if it gets into the cells, we get this activation of these drug pumps where they just pump the drug back out again. So that's one of the reasons why biofilms are so notoriously resistant to antimicrobial therapy. Mutations can also result in a change of proteins that serve as internal drug receptors. And that's particularly true with proteins associated with both the 30S and the 50S ribosomal subunits. Just remember there were a number of protein synthesis inhibiting antibiotics that interfered with peptide bond formation or translocation. And those proteins are part of the 50S or 30S ribosome. If the antibody can't bind to those particular ribosomal subunits, it's not going to affect those particular proteins. So random mutations in ribosomal proteins can confer resistance. Another mechanism of resistance that's been identified is sometimes microorganisms will develop strategies to protect drug targets. That's been the case in the development of resistance to the drug Cipro. Anybody remember what kind of a drug Cipro is, what it does? Cipro is one of those fluoroquinolines. And fluoroquinolines inhibit the DNA gyrase, or topoisomerase if you prefer. That enzyme that relieves the supercoiling in DNA as it unwinds. So the drug has got to be able to bind to those topoisomerase enzymes to be effective, to inhibit them. Resistant forms have been identified as producing a protein called MP1, and that actually binds to the topoisomerase enzyme. But it doesn't affect its function. But it prevents the Cipro from binding to the topoisomerase. So it's almost like a shield. Okay? The bacteria produce a protein that binds to the target, and that's a shield that prevents the antibiotic from interacting with the target. Sometimes bacteria will respond to drug therapy by lapsing into dormancy and tremendously decreasing their metabolic rate. Remember that drugs like penicillins, or most drugs that kill bacteria, will typically work the best during the log phase of growth, when the cells are actively metabolizing. because many of the strategies involve inhibition of metabolic reactions. So if you fall into dormancy, you're not going to be as effective. Some bacteria will respond to penicillin treatment by converting into an L form. If you remember or not, but L forms are forms of bacteria that sometimes during their life cycle have a cell wall and other times they don't. So if you lapse into the L form that doesn't have a cell wall, does it matter if you're exposed to penicillin? No. Penicillin inhibits cell wall formation, but you don't have a cell wall at that point in time. And then, again, if you can establish yourself as part of the biofilm, that's going to give you another whole set of strategies potentially that confer resistance. So there's a lot of things that are going on. So what can we do to try to minimize this particular threat? What are some strategies that we can do to try to prevent the rampant development of drug-resistant forms of bacteria? Well, one of the big ones is doctors are going to have a real responsibility. And they're going to have the responsibility for prescribing antibacterial drugs only when they're needed and indicated. Not for viral infections. There are antivirals, but antibiotics are not antivirals. They've done some studies, and don't get hung up on these particular numbers, but typically there's probably a couple 100 million prescriptions for antibiotics written per year. Estimates indicate that probably about half those prescriptions were written for diseases that were actually viral in nature. Ear infections are a big one. Colds are a big one. Sore throats are a big one. Every time anybody gets a sore throat, they go, I've got strep. I need an antibiotic. Probably not very often. Do you have strep? So what can we do? We need to be able to test and determine the nature of the infectious agent. Now, if you do have strep, you do need to be treated, because an untreated strep infection can result in some nasty secondary consequences. It can develop into a condition called scarlet fever, and if it's untreated, it can also develop into a life-threatening condition called rheumatic fever, where your heart valves become affected. So strep's not something you want to mess around with. But in the past, what doctors have been faced with is an increased patient load, a demanding public that wants results, particularly parents bringing their children in. They've been up for 2 nights because the child has been up and crying because they've got a sore throat. You want some sleep? Doc, give me an antibiotic. Or give my child an antibiotic. Okay. Well, in the past, the doctor's response has to pretty much have been, well, I'll take the throat culture, come back and see me in a couple days, and I'll give you the results. And then we'll dose based upon those particular results. Now, the development of things like rapid strep tests, where you can go in and get results in 30 minutes or less, take a throat swab. These are all monoclonal antibody-based tests, which are highly selective, but they don't require the growing and culturing of the microorganisms in petri dishes. They really give a very quick response and result. Have really facilitated the ability of doctors to make good decisions about when they prescribe antibiotics and when they don't. Probably about half of the people that are in hospitals that receive antibiotics or that go to doctors and get antibiotics have never had a culture taken. It's more likely you're going to get a culture taken in the hospital. But if you go to a doctor's office, very likely you won't have a culture taken. And if there isn't a rapid test for a particular disease like strep, then the doctor pretty much is guessing or covering themselves and saying, I'm going to prescribe in case it is a bacterial infection. If it's a viral infection, I think the kind of philosophy is, well, it's not going to hurt you. It might not hurt you, but it contributes to the problem of development of antibiotic resistance. So we really need to get away from this idea of prophylactic application of antibiotics. There are some instances where that is appropriate. If you've ever had a fairly invasive surgery, like a knee surgery or a hip replacement, those, if they develop infections, are very serious, and particularly if they're resistant to antibiotics. For some period of time after you've had those particular type of invasive surgeries, if you go to the dentist to get a cleaning, the dentist will prescribe prophylactic antibiotics. Okay. What happens when you get a cleaning? You get all that tartar, calculus, biofilm, whatever you want to call it, scraped off your teeth, right? Can the hygienist who does it prevent any damage to your gum tissue at all? You usually have a little bit of bleeding, don't you? So the bacteria in that biofilm can gain entrance into your body through those damaged gingival membranes. And then if those particular bacteria spread throughout your body, they can then infect these new surgical sites. And so there are some situations where prophylactic antibiotics are warranted and necessary. Another thing that you need to do to try to stem this tide of antibiotic resistance is, let's say you are diagnosed with a bacterial infection. And the doctor says, okay, I'm going to give you some penicillin. What's the doctor assuming? They're assuming that that bacteria is not resistant to that penicillin, right? But do they ever test that? Typically not. So conducting susceptibility tests, actually testing to see if the identified pathogen responds to the antibiotic that you are considering prescribing, is really an important step in minimizing resistance. Because if you give someone an antibiotic and it's a resistant form, it's not going to do any good. But then again, the use of that antibiotic will tend to select for overall increase in bacterial resistance to that particular drug. So it's almost kind of like giving it for a viral disease. So where we can pick up next time is talk about some of the susceptibility testing mechanisms that are available and how we can screen pathogens for susceptibility to certain drugs.